Melatonin is a hormone that's made in the pineal gland, and it's highly correlated with the body's sleep-wake cycle. During the day, the pineal gland is relatively inactive, but when the sun goes down, it begins producing melatonin. In humans, the brain production of melatonin peaks in the late evening, usually around 9 p.m., coinciding with the body's normal time for sleep. Melatonin levels typically stay high in the blood for around 12 hours, and with daylight the levels naturally fall to barely detectable levels. The exposure to light is a major factor in regulating the human sleep patterns. Light exposure stimulates the nerves that run from the eye to the brain to signal to increase the body temperature and the release of the stimulating hormones like cortisol. Darkness, by contrast, stimulates melatonin production, and it decreases body temperature. Low melatonin levels have been linked to insomnia, particularly in the elderly. In a clinical review, serum melatonin levels were reported to be significantly lower, and the time to peak melatonin values were delayed in elderly subjects with insomnia when compared with age-matched normal controls. Research suggests that melatonin supplementation improves sleep. A meta-analysis of 19 randomized placebo-controlled trials, including 1,683 patients, found that melatonin improves the sleep quality. It increases the total sleep time, and it decreases sleep latency. Other studies suggest that it enhances alertness after sleep, and it reduces the number of times that people wake during the night. A randomized, double-blind, placebo-controlled trial involving 125 children and adolescents with autism spectrum disorder who are struggling with sleep disturbances found 2 to 5 milligrams of prolonged-release melatonin tablets for 13 weeks increased their total sleep time and decreased the sleep latency and sleep disturbances. Subjects in the trial unsuccessfully attempted to improve their sleep with 4 weeks of parent-led behavior intervention alone prior to the trial. Circadian rhythms are biological processes, and they vary during the natural 24-hour day-night cycle. Maintaining consistent and healthy circadian rhythms promotes the overall health and well-being, including healthy sleep patterns. The human body contains 2 regulatory elements that respond to circadian rhythms: the central clock and the peripheral clock. Melatonin helps to modulate the central clock. Nobletin, a flavonoid derived from citrus peel, helps to modulate the peripheral clock. Preclinical research suggests the nobletin can protect against metabolic syndrome and age-related health deterioration, in part by helping to regulate the peripheral clock. When given to obese mice, the nobletin counteracted the metabolic syndrome and increased the energy expenditure. Another animal study demonstrated that the nobletin reduced the symptoms of delirium in mice. Researchers believe the mechanisms of action involve regulating the expression of the proteins that help to maintain a stable circadian rhythm. Nobletin can also help to alleviate circadian rhythm disorders and jet lag. L-tryptophan is an amino acid, and it serves as a precursor for serotonin and melatonin. L-tryptophan supplements can increase the amount of melatonin made by the pineal gland, thus facilitating sleep. L-tryptophan has long been of interest in the sleep field. Research dating back over 44 years found 1 gram reduced the amount of time that was needed to fall asleep. Like melatonin, L-tryptophan levels decrease with age. Therefore, L-tryptophan supplementation can help in the treatment of insomnia. Animal studies have found tryptophan supplementation reduced the activity at night, and it led to other biological changes conducive to sleep, such as lower core body temperature and reduced levels of interleukin-6, an inflammatory cytokine. In one small human clinical trial, an intravenous infusion of L-tryptophan caused dramatic increases in plasma melatonin levels, and it had a sleep-inducing effect, regardless of whether it was administered in the day or the night. In addition, the L-tryptophan can help to alleviate some forms of depression, which can exacerbate insomnia. An analysis of data from the National Health and Nutritional Examination Survey involving over 29,000 individuals determined tryptophan intake was positively associated with sleep duration. Another study examined whether the consumption of cereals fortified with tryptophan improved the sleep-wake cycle in 35 individuals aged 55 to 75. The study compared the consumption of either the subject's usual diet, cereals enriched with 22.5 milligrams of tryptophan per 30 grams of cereal, consumed at breakfast and dinner, or 60 milligrams of tryptophan per 30 grams of cereal, consumed at breakfast and dinner. The cereal with the higher dose of tryptophan increased the sleep efficacy and the total sleep time, and it decreased sleep fragmentation and latency. Another randomized study involving 33 male college students investigated the effects of tryptophan consumption at breakfast time and daytime light exposure on melatonin secretion and sleep. Subjects ingested either a tryptophan-poor meal paired with dim light, tryptophan-rich meal with dim light, tryptophan-poor meal and bright light, or tryptophan-rich meal with bright light exposure. The combination of bright light and a tryptophan-rich meal at breakfast promoted melatonin secretion in the evening and supported higher quality sleep. Glycine is a non-essential amino acid, and it transmits chemical signals in the brain. It helps to support bone health, digestion, and metabolism. This compound is also a novel and safe way to promote healthy sleep patterns. In one randomized, single-blind crossover trial, 11 healthy volunteers who took 3 grams of glycine 1 hour before bed experienced a shortened time to sleep onset and improved sleep satisfaction without next-day sleepiness. In a randomized, double-blind crossover trial, 3 grams of glycine before bed improved the sleep quality and the next-day fatigue. In a randomized, single-blind crossover trial, 10 sleep-restricted but otherwise healthy men were given 3 grams of glycine or placebo before bed. The next day, participants took memory, cognition, and reaction time performance tests and a questionnaire rating their fatigue level. The subjects that were given the glycine had reduced fatigue and sleepiness, and they'd improved neurobehavior performance compared to the control group. In an animal model, glycine administered to rats who've experienced disturbed sleep patterns resulted in shortened sleep latency, decreased body temperature, and the induction of non-REM sleep. Oral glycine administration may also help to elevate the levels of serotonin, a neurotransmitter that plays a role in mood, learning, memory, appetite, and sleep. Magnesium is a mineral, and it plays a role in cellular communication, energy production, muscle function, blood sugar maintenance, and the regulation of the circadian rhythms. The Food and Nutrition Board of the Institute of Medicine at the National Academy recommends a daily intake of 420 milligrams for men and 320 milligrams for women aged 31 and older. Optimal levels of magnesium are associated with normal sleep regulation. Magnesium deficiency is associated with shorter sleep duration, and it may cause inflammation, which could result in conditions associated with poor sleep, like diabetes and cardiovascular disease. In addition, research suggests as sleep restriction increases, intracellular magnesium concentrations decline. Magnesium supplementation combined with melatonin and zinc has been shown to improve sleep in the elderly. Another trial found magnesium supplementation helped to relieve insomnia related to restless leg. In a double-blind, placebo-controlled clinical trial, 43 patients in long-term care with chronic insomnia were given 5 milligrams of melatonin, 225 milligrams of magnesium, and 11.25 milligrams of zinc or placebo 1 hour before bed every night for 8 weeks. The treatment group saw improved sleep quality and improved quality of life measures when compared with a placebo. A form of magnesium known as magnesium threonate may be beneficial for sleep as it penetrates easily across the blood-brain barrier. Zinc, a trace element that's essential for many biological functions, including the immune health and neurocognition, can help to reduce the risk of inflammatory diseases, and studies indicate that zinc can also play a role in facilitating sleep. A literature review indicated serum zinc levels vary with the amount of sleep, and oral administration of zinc increases both the amount and the quality of sleep. In another trial, mice who were fed a yeast extract containing zinc had increased levels of non-REM sleep and decreased motor activity levels when sleeping. Another study showed that women with the highest levels of zinc in their bodies slept for longer periods of time than women with the lowest levels. As previously mentioned, when combined with melatonin and magnesium, zinc also supported the quality of sleep in the elderly. How zinc supports the sleep patterns remains unclear, but some researchers speculate that oral administration of zinc rapidly increases the blood levels, which activates a signaling pathway in the central nervous system. Zinc might act as a circadian regulator that induces sleep. Valerian is a perennial flowering plant, and it's been used for medicinal purposes, including inducing sleep, reducing anxiety and stress, and stimulating digestion since ancient times. Valerian root contains several compounds with sedative effects, including valerianic acid and valproates. Valerian root is believed to affect the transportation and the liberation of GABA, and it stimulates GABA receptor activity. In one randomized, triple-blind controlled trial involving 100 postmenopausal women who were struggling with insomnia, a treatment with 530 mg valerian root for 4 weeks resulted in improved sleep quality. One preclinical model examined the effects of a mixture of hops and valerian on sleep quality, and the researchers found the sleep behaviors, including the total sleep time, improved via the modulation of GABA signaling pathways in the treatment group. One study compared the effects of 600 mg of valerian to a commonly prescribed tranquilizer oxazepam. During 6 weeks of treatment, the valerian showed comparable efficacy to 10 mg of the drug. A review of relevant studies indicated the valerian is an advisable treatment option for people who are struggling with sleep issues, and it appears to have a favourable safety profile with no known interactions with common medications. The typical dose of valerian is about 300 to 600 mg, 30 to 120 minutes before going to sleep. It can take up to 2 weeks of daily use for the full sedative effect to manifest. Chamomile is a popular herb, and it's often used as a tea to promote sleep and relaxation. The dried flower extracts of the chamomile contain terpenoids and flavonoids, and these can help to combat inflammation, insomnia, ulcers, wounds, pain, and muscle spasms. Sedative properties of the herb are believed to be due to certain flavonoids that may modulate benzodiazepine receptor signaling in the brain. One randomized, placebo-controlled, double-blind study involved 57 people with a history of anxiety and depression, and it found chamomile supplementation reduced mood and depressive symptom scores. In a randomized controlled trial of 80 postnatal women, drinking chamomile tea for 2 weeks helped to alleviate the symptoms of depression and improve their sleep quality. In a randomized controlled trial, 200 mg of chamomile twice daily for 28 days improved the sleep quality in a group of elderly subjects. In another clinical trial of 77 elderly subjects, 400 mg of chamomile twice per day improved the reported sleep quality measures. One randomized placebo-controlled clinical trial found 270 mg of chamomile twice a day for 28 days improved the daytime functioning of 34 subjects with sleep problems. Passion flower, or Passiflora incarnata, is a member of the passion flower genus. It's been used to treat anxiety and insomnia in Europe, and the active compound appears to interact with the GABA and the opioid system. Extracts of this plant have shown sedative, anti-diabetic, and anti-asthmatic activities in preclinical research. In one randomized double-blind trial, 78 insomniacs were given either 10 mg of zolpidem or a combination of hops, valerian, and passion flower for 2 weeks. Both groups displayed improved sleep time, sleep latency, and night waking. One randomized study compared the supplementation with either passion flower or St. John's wort in 59 menopausal women who were experiencing sleep disturbances and depression. After 6 weeks, both groups demonstrated improved sleep quality. In one animal model, the Passiflora incarnata was shown to reduce anxious behaviour. Additionally, another animal model found Passiflora-derived compounds prevented diazepam dependence in mice when given with the drug over a 3-week period. While the extract has a good safety profile, more rigorous clinical trial data would be necessary to assess its efficacy in humans. Ashwagandha is an Indian herb and it's been used for centuries as a wellness-promoting tonic. It can be beneficial for reducing anxiety, promoting calmness and treating insomnia. The herb is best characterized for its effect on stress. A randomized, double-blind, placebo-controlled trial had 64 individuals who were struggling with chronic stress, and it found 300 milligrams of an ashwagandha extract twice a day for 60 days reduced the reported stress and the serum cortisol levels. A systematic literature review included 5 human trials, and it concluded ashwagandha supplementation at 125 milligrams up to 12 grams per day improved the self-reported stress and anxiety scores. In vitro research suggests a component of the herb, withanone, protects the neural cells from oxidative stress. Because emotional stress can be a significant contributor to insomnia, ashwagandha might help indirectly to improve the sleep by improving your response to stress. A randomized, double-blind, placebo-controlled trial published in 2020 purported to be the first to show that ashwagandha improved the sleep quality in human subjects. In this trial, 144 subjects with non-restorative sleep took 120 milligrams of a standardized ashwagandha extract or placebo once a day for 6 weeks. Those who took the ashwagandha reported a 72% increase in the sleep quality versus a reported 29% in those who took the placebo. Moreover, the group who were taking the ashwagandha showed improvements in sleep efficacy, total sleep time, and sleep latency. Finally, the quality of life improved in the ashwagandha group. Ashwagandha has also been found to directly improve sleep in animal models. In one animal model, sleep-deprived rats were given ashwagandha extract and they showed improvements in behavioural tests, indicating supplementation might help to mitigate the cognitive and memory impairments that are seen in insomniacs. These rats showed decreased cellular stress and apoptosis in the hippocampus. Lemon balm is a herb from the mint family and is traditionally used for its calming and anxiety-reducing effects. One randomized, double-blind, placebo-controlled study showed 600 milligrams of lemon balm improved mood and significantly increased the self-rating of calmness. Lemon balm has also been investigated for the treatment of sleep problems. Research indicates rosmarinic acid, uracilic acid, and olanoic acid are the active compounds in lemon balm, with rosmarinic acid being responsible for most of the activity that's associated with the herb. Lemon balm may inhibit the GABA transaminase, and this plays a role in anxiety and neurological disorders. In a randomized controlled trial of 11 healthy people who were supplemented with lemon balm, containing 500 milligrams of rosmarinic acid, the herb was found to be safe and tolerable. In an open-label prospective study, 20 stressed individuals took a standardized lemon balm extract for 15 days. Improvements in anxiety and insomnia symptoms were reported. In a randomized placebo-controlled trial, 100 menopausal women with sleep disorders received a combination of valerian and lemon balm. The treatment group experienced a reduction in sleep disorder symptoms. One study found a combination of valerian and lemon balm improved the symptoms of sleep disorder in 81% of children who took the preparation. Aromatherapy is an alternative medicine practice, and it utilizes plant oils to treat health problems. Lavender oil has been extensively studied for the treatment of insomnia. A randomized controlled clinical trial involving 79 college students with sleeping difficulties found inhaled lavender combined with good sleep hygiene practices were more effective than sleep hygiene alone in improving the sleep quality. In another trial, 67 middle-aged women with insomnia were randomized to either inhale lavender oil for 20 minutes twice a week for 12 weeks or to receive sleep hygiene education. The treatment group experienced an improved quality of sleep when compared with the control group. In a 4-week study involving 28 postpartum women who were struggling with anxiety and depression, subjects were either randomized into a group that inhaled a blend of rose otto and lavender essential oil, or were instructed to avoid essential oil use during the study period. The aromatherapy group had a significant improvement in depression and anxiety scores with minimal risk of negative effects. Studies have found lavender oil also reduces feelings of drowsiness after waking. In an animal model, lavender oil use for 7 days reduced the behaviors associated with depression and anxiety. Exposure to the oil has also been shown to decrease aggressive behaviors and to improve social interactions. Some researchers are examining the use of oral lavender to combat sleep problems. In a multi-center randomized double-blind trial, an oral lavender capsule was compared to lorazepam, a benzodiazepine, in patients with generalized anxiety disorder. After 6 weeks, the lavender was effective at reducing the anxiety symptoms with no sedative side effects. Lavender is thought to possess antioxidant abilities and to reduce inflammatory pathways. It also can alter ion channels in neurotransmitter junctions, which suggests that it might be beneficial in Alzheimer's patients. The oil can act via the limbic system and cause central nervous system depression when absorbed through the skin after topical application. While lavender oil appears to be safe and tolerable, and evidence supporting its use is promising, more clinical trials with long-term data will be required to further clarify its role in promoting sleep and relaxation. Saffron is a spice derived from the Crocus sativus flower, and it's been widely used as a flavoring agent and traditionally used to treat depression and asthma. It has powerful antioxidant properties, and preclinical studies indicate its major compound, saffronol, croquin, and quercetin, can have anti-tumor and anti-atherosclerotic properties. Animal and in vitro research suggests saffron might be a potential therapeutic agent for Alzheimer's disease, cardiac ischemia, and age-related macular degeneration. In vitro and in vivo studies indicate saffron and croquin may have neuroprotective properties. Preliminary evidence suggests saffron can promote sleep. Animal studies on saffronol indicate it supports non-REM sleep and it decreases sleep latency. One placebo-controlled human trial examined the effect of 300 milligrams of saffron on sleep quality in 50 diabetic patients. After 1 week, sleep quality improved in the treatment group. However, more high-quality human data is necessary to determine if and how the saffron can be used to treat insomnia. Hanukkah is a ligand extracted from the bark, seeds, and leaves of the magnolia tree. It's been traditionally used to treat mood disorders, including anxiety and depression, and it's believed to have anti-tumor, anti-inflammatory, and antioxidative effects. It can cross the blood-brain barrier, resulting in enhanced bioavailability, and research suggests it exerts neuroprotective abilities in the central nervous system. Hanukkah enhances GABA receptor activity in the neurons in the hippocampus. In one animal study, mice who were administered Hanukkah experienced shortened sleep latency and increased amounts of non-REM sleep. Several older research studies with animal models support these findings, including one where mice were given 7 daily treatments of 2 different traditional plant-based tinctures, and they had reduced anxiety and improved maze test scores. These effects were mainly due to the Hanukkah content, as varying the chemical content of the samples still produced similar results as long as the Hanukkah was contained in the sample. The Hanukkah-free preparations didn't influence anxiety levels in the mice. Mice who were administered 20 milligrams per kg of Hanukkah showed reduced anxiety levels in a maze test without a reduction in motor function. Another animal model demonstrated that Hanukkah had similar anxiolytic effects as diazepam, but without the possible amnesia and the drug dependence. While these results are promising, more human trials would be needed. Danshen is a herb traditionally used to promote sleep and proper blood circulation, thus improving cardiovascular health. The danshen, whose main components include rosmarinic acid and salvinoic acid, is often used in combination with borneol for better pharmacological results. Early animal research suggests the danshen has tranquilizer-like properties. However, well-designed clinical data is lacking, and adequate research must be carried out to understand how the danshen alters sleep. Certain peptides derived from the enzymatic breakdown of milk proteins might relieve stress-related sleep disorders. These peptides are believed to act on the GABA receptors in the brain, inducing relaxation and sleep. In one study, the bioactive peptides increased the time spent sleeping and reduced the amount of sleep needed after just 2 weeks of treatment. Another randomized, double-blind, placebo-controlled study on 63 women suggested 150 mg per day of milk protein alpha S1 casein hydrolysate reduced the stress-related symptoms, including sleep difficulties. For more on herbs, supplements, and natural treatment plans, check out my website.